During my research study as a PhD student in KISS University, I choose a topic on phytoplankton, and the full topic of my research work was water quality changes in relation to phytoplankton communities and rainfall patterns in River Malewa of Lake Naivasha Basin. And why I choose this topic is that I found out in the literature that most studies have not focused much on the rivers and streams, but unlike Naivasha or lakes, that there is a lot of information and knowledge about lakes. But I found out that little knowledge and few researchers that have studied in River Malewa, and the information is very scanty. I choose it because I found out this river has a lot of activities that go on. There's a lot of agricultural activities, urbanization around major towns that use this water. It's very important and vital to measure resilience or monitor this river, as it is very important in our nation. And it is also a river that feeds Lake Naivasha. About 90% of the water comes from River Malewa. And if we don't monitor this lake for some time, we may find that these resources become deteriorated, they become polluted, they might even become less water because now and then there are levels that decrease, and hence killing maybe the aquatic organisms that are in there. And this river, previous research that we did as chemistry scientists, we found out that there is this small fish called Babas amphigramma, that is a riverine fish. We also got the catfish, and there is also that largemouth trout fish that is found in this area. So this river is very essential, and it can be transformed in terms of aquaculture. When I was in Vietnam, aquaculture technology is very high because they produce a lot of fish in riverine aquaculture, which is not so common in Kenya. But I will recommend in River Malewa, all the stakeholders should be called upon to come together and come up with resolutions of food security. And one of the major remedy for our country is to go aquaculture. And I see this opportunity from the bridge of River Malewa towards 1 kilometer upstream. We can do a lot of aquaculture, and we can produce 1,000 tons per month, and which will substitute to our percentage in GDP of our country, that can increase our gross GDP for the country, and also improve livelihoods and also create job opportunities. With my technicians took to the field study, picking samples for selected nutrients. We were also doing physicochemical analysis, and that is to ascertain the levels and to see whether the river is well managed and protected. And during my study, we selected 10 sampling points from the river source downstream to the main lake, and took 10 sampling points where we used to pick samples for 1.5 years. And during phytoplankton analysis, bottles were pre-washed with 5% HCl, and then double rinsed with double deionized water and dried before picking the sample. In the field sample, we did phytoplankton sample collection by filtering 40 liters of water, and then through 30 microgram phytoplankton net, that thereafter we concentrated to 1 liter and then transported, and then filled with Lugo solution, then transported to the lab for analysis. In the analysis in the lab, we filtered the sample and then concentrated into 50 ml. Then from there we picked 1 ml into a acidic laughter cell. Then we observed the phytoplanktons that were there in that sample. We identified using a resolution of 40 times resolution. Most of the phytoplankton species that were major in this sampling period from 20 November to 2022 January, The major phytoplankton species that were common were Navicula species and Nabian species. We have Fragilaria, then we have Meridian, then we have Avisomenon, among others that were leading. In lake points, we saw Serratium species and Sigma species that were major common in highest frequency. But the major frequency in the river were the Navicula, Anabaean, Meridian, Fragilaria, and Avisomenon were very common, Cholera. And we have other species that were Acistrodesmus. They were very, very high in large numbers in this river. I request most of my listeners to read my article that is almost out and to see the gaps. Thank you.